Good morning. Good afternoon, actually. Thank you, everybody, for showing up. Thank you, Dr. Matthews, for the invitation, and Dr. Jeffers, I think, is in the audience, which is a little intimidating. We're going to talk about coccidiosis, but anyway, we'll try to do a fair job with it. Well, Dr. Matthews assigned me the disease, the diagnosis, and the lesion scoring. And like he said, I teach a whole week the Master of Avian Medicine graduate students at the University of Georgia. And I only spend about 8 hours on the disease, probably another 8 hours on lesion scoring between the two lectures, chickens, turkeys, and then the wet lab, and about a half a day on diagnosis. So we're going to try to do all that in about less than 10 minutes each. So you're going to get a few highlights, but I'm going to keep on moving because the time is running. So let's talk about the disease, first of all. This quote from a paper, a review paper written by Dr. Hawkins back in 1950, says coccidiosis is today a much studied, widely known, and little understood disease of poultry. Here we are 74 years later, and I can open exactly with the same statement. For example, some questions remain unanswered. Why do not the coccidia of chicken infect the turkey and other birds? What are the factors that make coccidiosis so resistant to environmental conditions? What makes sporozoites invade cells in specific segments of the intestinal tract? What makes sporozoites within the same location invade epithelial cells at the tips of the villi versus others the crypt? What causes the infection to be self-limiting? Why do not merozoites and schistons follow each other in indefinite succession as they do in Plasmodium? What determines that one second-generation merozoite will become a third-generation schison, a second will become a macrogamete, and still a third will become a microgametocyte? What determines that some third-generation merozoites will become macrogametes and others microgametocytes? So as you can see, we still know very little about the disease. As far as its relevance, for as long as we continue to raise chickens on the floor, chickens and turkeys on the floor in confinement with relatively high stocking densities, just like from the beginning of times of the poultry industry, coccidiosis has been and will remain one of the most important diseases of poultry. As far as the definition, the disease is caused by protozoan parasites of the genus Eimeria that infect different segments of the intestinal tract, causing destruction of epithelial cells leading to inflammation, increased permeability, malabsorption of nutrients, impaired growth, poor feed utilization, increased mortality, and increased susceptibility to secondary bacterial infections. As far as the types of coccidiosis that we have, well, of course, we have the clinical one, where we have definite symptoms of disease, such as diarrhea, bloody droppings, birds huddling under brooder stoves, etc. Subclinical coccidiosis, however, doesn't display signs of disease, but is very prevalent, and if you monitor for gross lesions and parasites, you will find them. And these will have adverse effects on performance, particularly in feed conversion ratio and daily weight gain. Yet coccidiosis is an old term, but this one is coined to reflect the mildest of the infections where there is no clinical signs and no measurable effect on performance. Since we use methods to prevent coccidiosis normally in commercially raised poultry, clinical coccidiosis is rather rare, except when we have overwhelming challenge post-vaccination in NAE or no antibiotics ever, or raised without antibiotics birds. Is usually something related to poor administration of the vaccine or poor cycling of the vaccine. We can have problems if there is severe immunosuppression or feed mill errors, such as leaving the anticoccidial drug out or feeding it at lower dosages. And of course, if we improperly handle vaccines and allow them to freeze or expose them to direct sunlight, we could have cocci problems down the road. Like I mentioned before, subclinical infections are the norm, and these are the hardest to recognize due to the lack of clinical signs or obvious lesions of disease. The flock appears healthy, but performance may be substandard. And this is the main reason why we conduct necropsy sessions or so-called cocci checks. So one important thing to remember is something Dr. Barnes taught me a long time ago. Coccidiosis is not a thief, it's an embezzler. With a thief, the damage is immediately apparent. You were robbed instantly. With the embezzler, things remain pretty quiet until you balance the books. In this case, when you settle the flock, you realize, whoa, what happened? 5 points worse feed conversion and 30 points less weight gain. There are 7 universally recognized species of chicken coccidia. These are listed here. The ones I highlighted with a black background, Acerbulana, Brunetti, Maxima, Nicatrix, Antonella, produce distinctive gross lesions that we can find in the GI tract. This round structure represents the gizzard. This represents the vent. So as you can see, Acerbulana infects the proximal small intestine, Brunetti the distal small intestine, the colon, the rectum, Maxima the middle small intestine, Mitis the distal small intestine, but doesn't produce lesions typically. Nicatrix, the middle small intestine. Praecox, another one that doesn't produce distinct lesions, or at least very minimal lesions. The proximal small intestine. And Tenella, of course, as everybody knows, infects the ceca. Molecular surveys conducted in any part of the world where they've been conducted always show the presence of the 7 species. Here, the orange bar is the United States, the blue bar is Brazil, the gray China, and the green Algeria. But as you can see, although the incidence varies among the various countries, we find Acerbulana, Brunetti, Maxima, Mitis, and in the next slide, Nicatrix, Praecox, Antonella. So the species are found. Now, I want to alert you about the new species, which most of you already know. Their practical relevance is unknown. Their economic impact unknown. They're not prevented by immunoprophylaxis with current anticoccidial vaccines. And they were, if you remember, for many years, we call them OTUs, Operational Taxonomic Units, and they were OTU X, Y, and Z. Well, since Blake conducted the molecular and biological characterization of each species, they have been named now Eimeria lata, Eimeria nagambiae, and Eimeria zeria, based on their initial location or place of isolation as far as geographical isolation. In a more recently reported paper done through next generation sequencing in a survey of coccidian nematodes in the state of Georgia, published by Terra and co-workers, University of Auburn, they show that the OTUs exist in the US. Here's the paper. If anybody's interested, I'll be glad to supply a copy. They did the genetic as well as the biological characterization of each of the 3 cryptic Eimeria species, or the OTUs, Operational Taxonomic Units, that I mentioned. And this is the paper, Avian Diseases, where it was published and reported that in addition, in these backyard flocks, or pasture flocks, they found operational taxonomic units, OTU, X, Y, and Z, although they have not been biologically characterized yet in the US. I'm not going to leave turkeys out because they're another important species in the US. And Coincidentally, there's also 7 recognized Ameri species in turkeys. Here, the ones I highlighted with a black background, adenoides, gallipobones, and meleagrimides, produce distinctive growth lesions in the intestinal tract. Same diagram, this is the gizzard, this is the vent. So as you can see, different locations, but the 3 pathogenic ones, adenoides, the ceca, gallipobones, the distal small intestine, colon, rectum, and meleagrides, the proximal small intestine and middle small intestine. The others, meleagrides, may produce some mild lesions. There's some debate. Dispersa, innocua, and subrotunda pretty much are considered non-pathogenic. The only molecular survey I found published is one from the US, one from Canada. And as you can see, in Canada, they found 6 out of the 7 species, and in the US, 5. In the US, we didn't find dispersa in this particular study. It was Midwestern turkeys, so it didn't cover the whole country. But in neither country found subrotunda in this particular surveys. So, talk about moving on to the life cycle. We're talking about a parasite, so life cycle is extremely important. These Eimeria or Coccidia have a direct life cycle, no intermediate host. The infected form is the sporulated oocyst. It's a self-limiting disease. Infection and immunity is species-specific. There are 3 phases well recognized: the sporogony occurring outside the chicken or turkey. The schistogony occurring internally is the first asexual round of multiplication. And the final one, gametogony, or the internal sexual multiplication phase. I'm going to very briefly mention the life cycle. This part of the circle that has been cut out represents the external phase, or the sporogony phase, or as I tell the students, the egg stage, or the oocyst. Starts with an unsporulated oocyst that has been passed on to the droppings into the litter. With the right conditions of moisture, temperature, and oxygen, they're going to sporulate and become infective. This happens within 24 to 48 hours, typically. And then the chicken is going to peck at the litter, pick up one of these infective forms, and through the mechanical action of the gizzard, this outer wall will be crushed, releasing the sporocyst, which through further action of the gizzard and the pancreatic enzymes, in particular trypsin and chymotrypsin, as well as bile salts, will dissolve the outer wall of the sporocyst, releasing the sporozoites. These will be swimming freely in the lumen, infect an intestinal epithelial cell, and start developing into a trophozoite 1, which has 1 nucleus, and by process of multiple fission is going to result in a multinucleated large structure contained within a cyst called the schizon or schizont. This is the schizon 1. When it gets to a certain, as the merozoites grow, eventually they're so packed in there that burst out, rupturing the cyst, swimming freely again in the intestinal lumen, looking to infect another intestinal epithelial cell, which they do. At that point, they become trophozoites, 2nd generation. And from 1 nucleus, again, by multiplication, asexual multiplication, result in a multinucleated daughter cell that will eventually grow the merozoites to a size that is too large to be contained within the cyst. That will rupture out, being released into the lumen, repeating the cycle again, but this time they're going to differentiate sexually. The macrogametocytes are going to become the equivalent of the ovum, and the microgametocytes are flagellated and motile and are the equivalent of the mammalian sperm. The microgametocytes will fertilize the macrogametocytes, then the wall-forming bodies will align and will produce the external and internal outer walls of the oocyst. The zygote or oocyst will be passed on to the droppings as an unsporulated oocyst to complete the cycle. Coccidiosis is a self-limiting disease. This is very important characteristic of this parasite. As you can see, they'll start with low numbers, eventually building up as time goes on. In this particular case, birds were on ionophore anacoccidials. But even with that, at 4 weeks, you get a peak in number of oocysts produced, and as immunity starts to develop, the numbers start coming down. The asexual phase of multiplication turns into the sexual phase, and then eventually the numbers go back down to very low numbers of oocysts being passed on to droppings. As far as diagnosis, moving on to diagnosis, this is from the chapter on protozoal infections in the textbook of diseases of poultry, where we authored the subchapter on coccidiosis with Drs. McDougall and Jenkins, who will be presenting later on. For diagnosis, basically, we identified the gross lesions at necropsy in addition to the macroscopic forms of the parasite, either in mucosal scrapings or fecal droppings. That's the practical way. Dr. Jenkins will spend more time telling you about the molecular diagnosis of coccidiosis. If I remind you again, there are 5 species that cause distinctive gross lesions in specific regions of the intestine. I already mentioned the species and the areas they infect. There are 2 species that cause not distinct gross lesions, or the lesions are so mild and transitory in nature that it's very difficult to catch them. Those would be mites and praecox, as I mentioned before. A presumptive diagnosis of coccidiosis can be made based on the detection of the typical gross lesions, and that diagnosis can be strengthened by microscopic examination of mucosal scrapings of the affected areas or suspected areas. However, I call attention to this quote by the father of poultry coccidiosis research, Dr. Ernest Edward Teaser, which published this in 1932 in the Journal of Science, saying, attention should be called to the common fallacy of differentiating species on the measurements of oocysts. That is not a reliable procedure because there's a lot of overlapping. Confirmatory diagnosis, I must admit, is rarely done. For that, why? Because you require submission of samples for isolation and biologic and or molecular characterization, in addition to tissue submission for histopathological examination. Both procedures require expense and time, and that's probably the main reason they're rarely done. There are 3 species that can cause distinctive growth lesions in turkeys. I mentioned those: adenoides, Zika, Gallipovonis, distal small intestine, colon, rectum, meleagrammitis, proximal and middle small intestine. There are an additional 4 species that are thought to be non-pathogenic or less pathogenic or cause no gross lesions or very mild or transient lesions. This would be meleagritis, but also dispersa, inocua, and subrotunda. The 3 species that produce nice lesions in turkeys, I mentioned adenoides, Gallipovonis, meleagrammitis. An intestinal lesion scoring system similar to the one developed by Johnson and Reed in 1970 was developed for turkeys by Gatti and co-workers and published in 2019. However, its application in the field has been hampered by the common lack of gross lesions in turkeys. So how do we diagnose coccidiosis in turkeys? Well, in the field, gross lesions are rather rare. So, like I said, going by gross lesions is not reliable in turkeys. From a practical perspective, the way you diagnose this is by routine microscopic examination of mucosal scrapings from different segments of the intestinal tract. So there's a lot of cocci checks in turkeys to detect coccidiosis. Of course, age is a big factor. It's usually a disease of young turkeys. 3 to 4 weeks is when you're going to find the most prevalent time for coccidiosis. Confirmatory diagnosis, again, rarely done because there's a lag time to get results, and there's an additional expense to do molecular studies or biological characterizations. or submit tissues for a histopathological examination. Getting to the end of my talk, moving to the lesion scoring. The method developed by Johnson and Reed was actually developed for this purpose, which is Dr. Math is doing research studies and examining lesions. However, we, like I'm doing here in the field, and this is what I did for the last 27 years of my career, was going through intestinal tracts and applying the system of Johnson and Reed for the routine monitoring of coccidiosis. This is the landmark paper, probably in my opinion, the most cited paper in the coccidiosis world, and it's titled Anticoccidial Drugs, Lesion Scoring Techniques in Battery and Floor Pan Experiments with Chickens by Joyce Johnson and W. Malcolm Reed. Both are, unfortunately, have departed this life, but they both work in the poultry science department. Dr. Reed, been very truthful one time, told me, this is the only paper where I regret having given my master student the senior credit for the paper. But I told him, I consoled him. I said, it sounds better Johnson and Reed than Reed and Johnson. But anyway, and I know it's hard for you to believe that this was me back in the late 70s, early 80s, and this is, of course, Dr. Malcolm Reed. I was fortunate to spend the first 6 months of my stay in Athens, Georgia, in his home. Oops. Okay. So what are the lesion scoring? There are 4 regions by this system for which you score. You never score by species, although we do that in the field all the time. You score by regions. The upper region is the duodenum, proximal jejunum. The middle region is the mid-jejunum and the distal jejunum. The lower region is the ileum and the colon and rectum. And of course, the cecal region is the ceca. This is taken word by word from Johnson and Reed. This is what I use with the students at the MAM program. I'm not going to read these to you, but I'm just going to point out that the system goes on a scale from 0, where there is no gross lesions and obviously no effect on performance, to a maximum of +4, where you have the most severe lesions, or dead chickens or turkeys, depending on the case. And of course, you have a significant effect on performance. So these 3 slides came from the master himself, Dr. Reed. He didn't have a good 2 for some reason, but so I put 1 and I found in the field. So there's your 1, 2, 3, and 4, increasing degrees of severity going from 1 to 4. For Eimeria maxima, same type of scale, 0, no lesions, +4, the most severe lesions or mortality. And I took these from our Pfizer book, the Coccidiosis manual from Pfizer, developed years ago. And again, maxima +1, +2, +3, +4. As you can see, the degree of severity of the lesions increases as the score increases. Moving on to Nicatrix, same score, 0, no lesions, +4, the most severe lesions, or a dead chicken, or chicken in this case. As you can see, +1, +2, +3, +4. Brunetti, likewise, same scale, 0, no lesions, +4, the most severe lesions, or a dead chicken. And again, from the Pfizer technical manual, +1, +2, +3, and the most severe lesion, +4. Finally, Tenella, again, same scale, 0 to 4, 0, no lesions, +4, the most severe lesions, or a dead chicken. And as you can see here, that came from Dr. Reed. The rest came from the Pfizer Coccidiosis manual, +2, +3, and the most severe, +4. And finally, lesion scoring for turkeys. Again, like I mentioned, Gaddy developed a similar score going from 0 to +4, the most severe, or a dead turkey. You can see a normal ceca, 1 with lesions of +1, a few petechial hemorrhages, some foaminess, +2, more thickening of the wall, formation of some cores, +3, more thickening of the wall, more larger cores, and +4, a very thickened wall with a full core filling out the entire cecal pouch. Gallipovonis, again, 0 to 4, with 0 no growth lesions, plus 4 the most severe lesions. And finally, Gallipobonis going normal ceca. You can see this is distal small intestine. You can see the lesions there and more increasing lesions on the +2, +3, more severe lesions with some necrosis of the mucosa and beginning to form cores even in the ceca, and the +4, more severe necrosis of the mucosa of the distal small intestine and more exudate and core forming into the ceca. Meleagromitis, same scale, 0, no growth lesions, +4, the most severe lesions. And again, from the same publication by Gatti, normal duodenal loop, 1 with a score +1, where you see a little bit of exudate. +2, you see more exudate, more thickening of the wall. +3, more thickening, more exudate. Some ecchymotic hemorrhages, definitely here. And the +4, a very necrotic mucosal surface in the duodenum, with a lot more exudate accumulated. So, in conclusion, for poultry raised on floor in confinement, coccidiosis will remain a very important disease. Coccidia are monoxenous parasites that have a direct but very complex life cycle. In general, immunity is species-specific. There are a few exceptions. Infection is caused by ingestion of sporulated oocysts. Severity of infection is directly related to the number of sporulated oocysts ingested. Coccidiosis is a self-limiting disease. Subclinical infections are the most prevalent and the most damaging due to their silent nature. In the field, coccidian infections are diagnosed by the presence of growth lesions or observations of parasites in mucosal scrapings or droppings. Lesion scoring for coccidiosis was developed for experimental infections. However, the system was quickly adopted by the industry for the diagnosis of coccidiosis in the field. However, the system has proven to be more useful in chickens, where growth lesions are far more common than in turkeys. And with that, thank you for your attention.